Hi, everybody. Thank you so much, wherever you are in the world, for joining us to celebrate aquatic fungi diversity today on International Day of Biodiversity. I'm Moni Bohm. I'm based at the Global Center for Species Survival, and it's a great pleasure to say that we've got a wonderful set of talks lined up for you today to shed a light on these often overlooked, well, mostly overlooked, quite frankly, particularly in conservation, organisms that are so fascinating. A quick little bit of housekeeping. During the presentations, we'll kind of leave some time for questions after each of our speakers. Please use the Q&A in your Zoom to post any questions that you have to the speaker. The team here will monitor the Q&A section and pose your questions to the speaker after the presentation. The webinar is also being recorded, and we will then share the recording as soon as it becomes available afterwards with you. And then obviously feel free to pass this on to your friends, colleague, family, anybody you meet in the street so that they can learn about aquatic fungi too. Okay, let's start with a quick introduction of the hosts of this webinar. As I said, I am based at the Global Center for Species Survival. The Global Center for Species Survival is a partnership between the Indianapolis Zoo in the US and the IUCN Species Survival Commission. And the purpose of our center is to support the conservation efforts generally worldwide, but specifically via supporting the work of the thousands of volunteer experts that give their time to the IUCN SSC specialist groups, like, for example, the Aquatic Fungi specialist group that you will hear about shortly. For this global center, we have a team of coordinators who look after different species. We're actually currently waiting for our new fungi coordinator to start very soon, and are actively recruiting for a plant coordinator to complete the team setup again. And I will now pass on to, I believe, Laura.
Yes, that's me. I'm Laura Garzoli. I'm the Deputy Coordinator of the project FunAction, that is financed under the umbrella of the Biodiversity Plus EU call, and it aims at the conservation of aquatic fungi throughout Europe. And we have three bases for our work. We are working to create the foundational knowledge that we need to begin the conservation of aquatic fungi. We are committed to translate that knowledge in conservation planning, and we are working very hard to communicate about aquatic fungi as of today. So next slide, please. We are a very large group of enthusiastic people about aquatic fungi working across Europe and US. We are achieving wonderful milestones. So if you're interested in our project and if you want to know more, please browse our website and get in touch with us with our social media. Thank you.
Well, and that brings us to most fun. I'm Andy Bruder, one of the coordinators of also Biodiversity Plus project, Most Fun. The other coordinator is Teppo Rämä, that you see somewhere here. I see him here, but yep, depends on your screen configuration. And the ultimate goal of Most Fun is to add aquatic fungi to routine biodiversity monitoring programs. And to do this, we are collaborating with programs and projects that do biodiversity monitoring, but for other organism groups, but which might have samples, data, and experience that are relevant for aquatic fungi and can be used and reanalyzed for aquatic fungi biodiversity monitoring. We also contribute to and use existing concepts like essential biodiversity variables, but add aquatic fungi to them. We also do some of our own sampling to generate data, and particularly in understudied ecosystems like glaciers and estuaries. If you're interested in contributing to this, so as one of the stakeholders, maybe as one of the programs and projects, you will find the QR code down here on the slide that brings you to a survey. And on the survey that helps us to get to know our stakeholders or our contributors and collaborators to this project a bit better. So have a look there. There's also other stuff that you find on the link tree. I should also say that to help us with stakeholder interactions, we will develop a knowledge to action hub for aquatic fungi, and this will help us in these interactions, but also it will help us to expand our findings from the projects to other regions and beyond the project lifetime. Thank you for your interest, and that was from Most Fun. Get in touch with us if you're interested and want to know more.
Thank you. And this brings us to the last co-organizer of this event today, which is the IUCN Species Survival Commission Aquatic Fungi Specialist Group. This is a new group from the SFC created towards the end of 2023. We are now a group of almost 25 people from all around the world, covering different groups of aquatic fungi. And our vision is to develop the scientific and coordination basis for the first conservation assessment, planning, and action efforts for aquatic fungi. You have also there the QR code or the link to Linktree, where you will find connections to our social media and to our website. Get in touch with us if you want to know more about the work we are developing. And with no further delays, I will present our first speaker of this webinar. And our first speaker of today is Dr. Patricia Velez. Dr. Patricia Velez earned her PhD in 2014 in the area of biological sciences from the National Autonomous University of Mexico in Mexico for her work on marine fungi. Then Dr. Velez performed 2 postdoctoral stays at the National Autonomous University of Mexico and also at the Ensenada Center for Scientific Research and Higher Education, studying molecular ecology of freshwater and deep sea fungi, respectively. Currently, Dr. Velez is a faculty member, research professor at the Institute of Biology at the National Autonomous University of Mexico in Mexico, where her work aims to explore the diversity, ecology, and potential utilization of fungal communities, particularly in marine and threatened ecosystems. In all her work between teaching different courses and training several graduate and undergraduate students in different topics. There is a lot of work here, but a very special and recent addition also to the IUCN Specialist Group on Aquatic Fungi, which we are very happy to have Dr. Patricia with us. And that's it. I hand over to you, Patricia. Please, you can share and start your presentation. Thank you.
Thank you very much, Isabel, for the kind presentation. And thank you for the invitation. I'm delighted to be here with you and share some of our work and results on the exploration of ecological patterns and threats of marine arenicolous fungi. So, well, the oceans are the flow over nearly 3/4 of our planet, and they hold most of the water on it. They are also very important for the production of oxygen and also represent a very important sink for carbon dioxide. So we can all agree that the oceans sustain also the productivity of a number of countries, especially those coastal or island nations. So here the sustainable utilization and conservation of the oceans is very important for the conservation of global economic growth, but also for food security. However, we do need to pay special attention on the need to generate baseline data on marine underappreciated species. And here I put an example of fungi. We need to generate information on the levels of diversity at both taxonomic but also genetic level. We need to understand the distribution of these species, and also we need to understand what are the responses toward stressors. So here we go into marine fungi. This is an ecological rather than taxonomical group of species that have evolved, have adapted to grow and sporulate under marine conditions. They commonly occur as saprobes, therefore they play a very important role in nutrient cycling in the oceans. However, we do know only a small fraction of their diversity. Presently, we have described less than 1% of the overall fungal diversity comes from the oceans. And very specifically, arenicolous marine fungi are those marine fungi that occur in coastal beach sands and can be defined based on their unique specificity to the intertidal and subtidal region of the beaches. Here you can see they are really beautiful. They have a wide range of morphologies. So, well, first of all, marine sandy beaches, they give a number of how do you say, sorry, of benefits to humans when ecosystem is functionally correctly. So here I listed several examples. For example, they give economic spillover. They also are important for sediment storage and transport. They are also very important in buffering against extreme events, for example, storms or tsunamis. They also play a very important role in nutrient mineralization, water filtration, and so on. However, these very beautiful ecosystems also host economic activities. So anthropogenic and natural disturbances in sandy beaches act together. And the understanding of the effects of these two kinds of disturbances is poorly understood. So marine fungi in sandy beaches are inhabiting fragile systems. And this is very poor, it's actually poorly understood. We do know that marine fungi in beaches are susceptible to anthropogenic pressures. For example, they are very susceptible to changes in pH, temperature, nutrients, salinity, and so on. So we could expect that the reductions in the biodiversity of this group of fungi may lead to changes in ecosystem functional capacities. And here I included a map on the vulnerability to biome shifts due to climate change. And you can see that most of the coastal areas are actually in red, which represents a very high risk. So disturbance may be approached in two different ways. First, disturbance may be understood in terms of survivors, leading to catastrophic disturbance, for example, the explosion of a volcano. But also, disturbance, when understood in terms of survivors, may be non-catastrophic, for example, hurricanes or storms. And on the other hand, if we study disturbance in terms of the size of the disturbance itself, we may have sustained press, for example, anthropogenic disturbances, or episodic pulses, for example, hurricanes and storms. And this is very important in terms of ecosystem functioning because catastrophic disturbances usually leads to the directional replacement of species, which leads to species adapted to colonizing younger sites. Whereas non-catastrophic disturbances plays a very important role in species and diversity maintenance. On the other hand, sustained press disturbances eventually drive to species extinction, whereas post-disturbances increase biodiversity. And this is explained by the intermediate disturbance hypothesis. And this hypothesis says that diversity is higher at intermediate levels of disturbance when up, because under these conditions, opportunistic and competitive species can coexist. So now I'm going to share with you some data from coming from the Caribbean Sea in Mexico. And fortunately, we were conducting long-term monitoring when two very important hurricanes came to the coast. And you can see here in this graph that over the years, we've been recording a gradual loss of abundance, marine fungal abundance. around 32%. In terms of diversity, the pattern is the same. You can see that diversity has decreased over 2 years in 30%. And this agrees with data from a larger scale monitoring. You are seeing here data from 2000 to 2009, almost 10 years. And we can observe here that maybe we are losing rare species, and these loss may be irreversible. In terms of natural disturbances, on the other hand, you can see here in blue the rainy season, and during this rainy season, abundance of marine fungi increased by 80%. And this may be related to the influx of several detritus to the beach, leading to the proliferation of sand mycobiota. And in terms of the hurricanes, you can see here a slight increase after the hurricanes, and this is also linked to the intensity of the hurricanes, but we did not observe a clear pattern, not as clear as the one I just described with seasonality. So more evidence coming from the Gulf of Mexico, where we are having oil industry, port complexes, tourism, polluted river mouths, and a number of anthropogenic stressors. We conducted a sampling covering the entire Mexican coastline for the Gulf of Mexico. And you can see here the bar plot. You can see here that low diversity sites here and here are related to polluted river mouths and also very big port developments, but also the oil industry. Whereas the high diversity sites agree with the neighboring well-preserved systems. And this may be because fungal diversity is also related to plant diversity. So these ecosystems represent a varied source of substrata that provide an heterogeneous conditions for marine fungi to develop. So in conclusion, we observed no effect of hurricane disturbance on arenicolous fungi, but this fungi may be actually adapted to this natural disturbance in beaches, but we observed a clear dry storm season pattern on diversity and abundance, which increased during storm season. Hurricanes and storm season act as pulse disturbances that create patches of increased resources ability to marine fungi. And the effect of human disturbance on sand marine fungi is very negative. It leads to species loss, especially of those that are rare. So with this, I'd like to thank my group, my students, my colleagues, and I'll be glad to take any questions. Thank you.
Thank you. Thank you so much, Patricia, for this introduction to this super interesting group of fungi. I remind everyone that if you have questions for Patricia, you can put them in the Q&A. While maybe people are still a bit shy, maybe I can put you a question. Patricia, I guess for many people, every time they are on top of sand, they don't really think about having fungi below their feet. So maybe I would ask a more simple, general question. How many species can we actually find in one particular location? And do you have any special species that you are very fond of?
Well, I'll begin with my favorite species because I really do love this species. It's Coralaspora maritima, which is a cosmopolitan arenicolous marine fungus. It also produces interesting antibiotic. It also has been able to degrade several hydrocarbons, so it's a very interesting species. And answering to your first question, it depends on the group of marine fungi, because strict arenicolous fungi in one site, for example, 1 kilometer, a beach of 1 kilometer, for example, it depends also on the disturbance level. If you have hotels or any kind of human activities going on, but In Mexico, at least, I would say 22 species, maybe. If you have a well-preserved site and you have algae, mangrove, well-preserved neighboring ecosystems. So, yeah.
Super. So for everyone, next time you go to a coastal area, just remind that below your feet there is a whole range of aquatic fungi, that live there and make their magic happen. Let me see if we have questions here from the participants, and we have. So we have one question that says, what is the best way to preserve samples for marine fungal diversity analysis? More technical question.
Yeah, the best way, you mean in the lab? So if you choose a culture-dependent strategy, well, you need to preserve the isolates, fungal isolates. That's a way of doing it in a culture collection. There are many culture collections that host marine arenicolous isolates, actually. That's one way. But also you can preserve sand samples in the lab, dried sand samples. And in my experience, if the samples have developed ascomata, that are the reproductive structures that attach to the sand grains, after 7 or 10 years, more or less, you are still able to isolate the fungi from these dried sand samples.
Wow, that's quite good timing for that.
Yeah.
Thank you. We have another question on the Q&A from Jason, and says, you focused on Ascomycota. Do you know whether there are also arenicolous parasitic fungi, example chytrid?
Yeah, there are other fungal phyla. Actually, we also have very few Basidiomycota, I would say, and also chytrids. Bad news is that chytrids are less studied in relation to Ascomycota. So we have no as much information on this phylum. But some papers have approached the diversity of this group using culture-independent strategies, for example, next-generation sequencing and so. But well, yeah.
Okay, and maybe I'll take maybe one last question here. I know there are other questions also popping in our chat, so I will ask, please make your questions on the Q&A. It's easier for us to follow, and also then our speakers can also write to you back an answer if they cannot answer live. I will make just one last question here. Is there any difference between aquatic samples and sandy beach samples? This zone is a particular habitat still not well categorized, not totally aquatic nor terrestrial.
Hey, that's a super interesting one. Yeah, actually, sandy beaches are the interface between marine, aquatic, and terrestrial systems. So it's an ecotone where environmental conditions change gradually, depending if you go into the ocean or into the terrestrial area. So in this very special ecosystem, several ecological groups of fungi coexist. Today, I only talked about true marine arenicolous fungi, which are endemic to these specific conditions. But you can also observe terrestrial species or aquatic species, maybe those that infect algae or other marine animals. So yeah, you have several ecological groups living together in the sandy beaches, yeah.
Thank you. Thank you so much, Patricia. For the sake of time, we need to move to the next speaker, not species. Same species speaking, but a different speaker. We will try to answer the questions that are still remaining in the chat. Vani, I hand over to you to present our next speaker. Thank you.
Thank you. Wonderful. Yes, we'll do our best to get at least most of your questions answered. The next speaker is Professor Juri Nascimbene. Now, Juri is a full professor at the University of Bologna in Italy, and he is also the prefect of Botanic Garden and Herbarium of the university. And this is probably a word I'm going to stumble over, he's president of the Italian Lichenological Society. Done that very well. He has a PhD from the University of Trieste, and his research focuses on, well, a large number of things. Lichen taxonomy for one, and species inventories, vascular plant biodiversity patterns under global change, particularly in things like agroforestry systems, high elevation areas, and alpine forests. And I think he's going to take us into the realm of aquatic lichens, I believe. Is that true? So I'm very excited. Over to you, Juri.
Oh, thank you very much for this presentation. And actually, my presentation is focused on freshwater lichens, which represent a research line of which to which I pay effort. Let's go to the presentation. Okay. I share my video here. Okay. Yes, when I was asked to take this short seminar summarizing the characteristics of freshwater lichens, I was inspired by this famous title of the poetry by the Italian poet Petrarca, who referred to these chiare, fresche et dolci acque that perfectly summarize what is the top habitat, what are the top habitat conditions to find high diversity of these lichens. And we will see during the presentation how these lichens are actually related to clear, to cold, freshwater habitats. And let's pass from the habitat who inspired the poet, that where love reigns, to the habitat for lichens. But first of all, we can ask if actually there are lichens that live in fresh waters. We perfectly know that lichens are related to every type of terrestrial environment, even if they are extreme. We can find lichens, for example, in polar regions, in high elevation ranges, in hot and dry desert area. I think that everywhere you go in terrestrial habitats, you will find some lichen. But what's the situation in freshwater habitats? And actually, the situation is in this habitat we have only a few lichens that are adapted and that can establish permanently. And we have to imagine as we have a great environmental filter in which all the lichen diversity is filtered. We know that at the global level we know more or less 20,000 of species. But as you can see, when they pass across this environmental filter, we can find only a few species adapted to freshwater habitats. And for example, in temperate regions of the northern hemisphere, we know only 10 species that are truly aquatic and 50 at the global level, and around 200 of species that are in some way related to freshwater habitats at the global level. And mainly all these lichens, they belong to the group of Verrucariales. But what's the problem for a lichen to establish in freshwater habitat? The problem are many, and I have here I present you here a list of the main problems. So in relationships with respiration and photosynthesis, related to the the problems related to the diffusion of gases in water, light availability for photosynthesis, substrate stability, that is both superficial stability and stability of the stones on which lichens can establish. And another problem is related to the frequency and the length of inundation that may influence which species we can find in a given site. And we have also to consider the mechanical impact of water flowing that can reduce the potential of the thalli to develop, to establish firstly, and then to develop. And under these critic conditions, several lichens, even belonging to distant phylogenetic lineages, have evolved similar responses to address this kind of problems. So we have similar morphologies, similar partnerships in terms of photobionts that are included in the lichen symbiosis, similar ecophysiological adaptation. And in this way, different lichens address similar problems, but they create problems to us when we want to study them, since we have only a few morphological characters, for example, that are useful for species circumscription. And actually, we have to be aware of the fact that when we study freshwater lichens, we expect to find many cryptid species. Let's see some example on how freshwater lichens address the problem of living in this selective habitat. Most of the species, for example, to reduce the impact or to counteract the impact of water flowing, have a crustous thallus. And not only crustous, but if we consider two aquatic species, they have even a subgelatinous thallus that offer a very smooth surface that can reduce the resistance to water flowing. And again, problems with water flowing that are addressed by constructing structures, robust structures that can be observed here around the fruiting bodies. Like in the case of Perithecea, most of the species, we have to consider that most of freshwater lichens are Verrucariaceae, and so the fruiting bodies are mainly Perithecea, and these Perithecea are surrounded by these very robust structures that are involucral, that is the involucrellum. So many species have these strategies. And if we consider, as I already said, a given river, we have to consider that we can have during the year, especially in the Mediterranean, strong water fluctuation, and therefore the inundation length and frequency may vary depending on seasonal factors and environmental conditions in general. And this is reflected in the establishment of different lichens at different level from the water surface. As you can see in this picture, we can often observe a clear vertical zonation of the species, with the more aquatic species that are more related to this part here near the water, while in this area here that usually is larger than the previous one, we have amphibian, what we can call amphibian species that are adapted to irregular inundation, on only not permanent inundation, And while over this line, you can observe typical terrestrial lichens, also indicating that at this elevation, in this case, water never arrived. And in this perspective, freshwater lichens can be also used as indicator of fluctuation in water flow capacity. And let's see the problem of light. In general, these lichens love very much open environment. So their diversity maximum is like in environment like this in this picture, and not, for example, in lowland, inside lowland forests like in this picture. But when we speak about light, we have to consider also the light that actually reach the talus, and this also may depend on the sediment that are suspended in the water. And in general, they, as I previously said in the title, they prefer clear water with small amounts of sediments. But for maximizing the use of light under the water, several lichens also have internal adaptation that are different if compared with terrestrial species. And for example, in aquatic species, we have a high photobiont microbiont ratio. If we observe this example here, we can see a very huge layer of algae that is more than 3 or 4 times higher than in a normal lichen. And we can see also that these algae are well ordered in columns. And this probably is, this order is probably, but we actually do not know for sure, maybe related to a more effective management of the light inside the talus. And yes, the other ecological factor important for freshwater lichens is temperature. And actually they love very much cold water, the so-called fresche acque, the cold waters of the poet. Since under this condition there is more solubility and diffusion of the gases. And actually, in this perspective, we have to expect top diversity, for example, in mountain and subalpine environments, while in lowlands, we can find these lichens only in microrefugia, like, for example, springs, that offer relatively cold water, or in shaded condition. But we know that in shaded condition there is a threshold with light requirements. So top diversity in open and cold environments. And you can imagine that all this stuff has implication for global change. For example, increasing temperature, changes in water supply, changes in water body level, especially in the Mediterranean, and changes in trophic level. We have to think about eutrophication, and sediments have implication or can be considered threats for this lichen biota. And this is a lichen biota which needs a lot of effort to be better explored. We know not so much. Our knowledge is very limited, is very scattered. We do not nothing, or we have small information on their actual diversity, distribution, ecology of the species, ecophysiological adaptation, and so on. And every year, several species are continuously described. as for example, in this case from the Mediterranean. Last year we described the 3 species, but this is only an example, since recently colleagues described several species from the Nordic countries, for example. And so this is a group of the lichen biota that still needs much effort in terms of exploration of species and their distribution and ecology. And under these circumstances, it's difficult to say something about conservation, obviously. And the only thing I can suggest is that we have to put effort to strictly protect the top habitat for these lichens, the clear, the cold, freshwater habitats like those represented in this picture, for example, that is taken in the Dolomites. So strict habitat protection of these rivers, of spring habitats, would protect the diversity of freshwater lichens until we do not know some more about their distribution and ecology. Thank you very much for your attention.
Thank you so much, Juri. That was super fascinating. We've got 2 questions in the Q&A. In the interest of time, we're doing one live. Please, let's keep the answer relatively short because we're running behind. Kay is asking, from my experience, lichens prefer to dry out during periods of metabolism. How have these lichens adapted to a high moisture environment, and how do they fight against high moisture preferring pathogens?
Yeah, I spoke about some morphological adaptations. We have also to consider that the gelatinous thallus that characterize many freshwater lichens or typically aquatic lichens may also be a tool for maximizing the gas metabolism inside the thallus. But we have also to consider the partnership. I had not the time to speak about this, but while most of the algal partner are in the Trebuchetacea, if we consider terrestrial lichens in general, here we have some specialized algae that are more adapted to freshwater habitats in terms of their ability of maximizing photosynthesis in under submerged conditions. So there could be some additional adaptation that we, however, do not know. For example, it would be interesting to explore better the meaning of this of the structure of the algal layer that we find in different species that are also phylogenetically not so strictly related. And it would be interesting to know how this work, which is the mechanism. But I am hunting for species, but not in the ecophysiology of these lichens. Yeah.
Wonderful. Thank you so much, Juri. There's a few more questions in Q&A that we'll get to type answers to as much as possible. Isabel.
Thank you, Mani. And it's my pleasure to introduce the next speaker. The next speaker is Juliana Barros. Juliana is a PhD candidate in biosciences at University of Coimbra in Portugal, affiliated with MARE, Marine and Environmental Sciences Center. Juliana researches, in her research, she investigates the ecological impact of emergent contaminants, including nanoplastics and metals, on freshwater fungi. By studying the effects across multiple levels of biological organization, from ecosystem processes to molecular responses, her work seeks to contribute to conservation efforts and pollution management strategies in freshwater ecosystems. Thank you so much, Juliana, and I hand over for you for your presentation.
Okay, good afternoon, everybody. I'm very happy to be here today. Thank you so much for the invitation. Let me see if I'm already sharing my screen. Okay, not yet in presentation mode, right? So let me put here. Okay. So, okay, as I said, I'm super happy to be here today to celebrate the International Day for Biological Diversity, discuss a little bit about these amazing organisms that are the aquatic fungi. And today I'd like to discuss a little bit about their importance in freshwater ecosystems and how they can be employed as a tool in a multi-scale, as a multi-scale bioindicator for freshwater ecosystem health. So first of all, let's understand bioindicators. What are they? They are organisms or biological responses that reflect the state of the environment. They are important because they can detect both early signs of ecosystem stress, but also cumulative impacts, and therefore they are essential for supporting ecological risk assessment. While many organisms have been widely employed, and although even some fungi have also been used, they are often overlooked, despite their key ecological roles in the ecosystem. So what is this role? In streams, organic matter decomposition is driven primarily by aquatic fungi, specifically aquatic phomycetes. This is a crucial process responsible for the nutrient cycling in fresh waters and the transfer of energy to higher trophic levels, consequently maintaining the ecosystem integrity. But these organisms, they are very responsive to environmental changes and stressors, what makes them effective candidates for monitoring freshwater ecosystem health. One of their greatest strengths is their ability to indicate stress across multiple biological scales, from molecular biomarkers to ecosystem level processes. This multi-scale sensitivity allows us to capture both subtle and severe forms of environmental degradation. So how can we do it? To capture these effects, we can combine traditional methods like litter bag experiments, microcosms, but we can also rely on advanced technologies and molecular approaches, such as qPCR, DNA, RNA sequencing, and metabarcoding, etc., which give us room to study these fungi at various levels. Now, talking a little bit about these levels. So at the molecular level, for instance, new technologies allow us to quantify the production of stress-related metabolites like the glutathione. We can assess alterations in their enzymatic activity, for example, the lacase that tend to increase under metal or pesticide exposure. We can observe changes or impairments in DNA, also analyze gene expression patterns that can serve as early biomarkers of stress. At cellular responses, we also find various indicators, starting from the fungal cell wall, for example, that act as a first barrier, often binding or blocking contaminants' entrance. If not enough, these contaminants can damage the plasma membranes, disrupt organelles' functions, for instance, the mitochondrial function, triggering the production of reactive oxygen species, this oxidative stress. And these effects can be, for instance, easily visualized using a confocal microscope. Here, for example, we see the confocal microscope images of the fungi mycelia. The red areas in the first image indicate the plasma membrane damage, while in the second image shows direct oxygen species production. The more intense the red, the greater the impact. And as we move to the bottom images here, you can clearly see an increase in red signal with the higher concentrations of contaminants. In this case here, we were analyzing the impacts of copper along with nanoplastics. At organism level indicators, we can observe, for instance, differences in growth patterns and biomass production. Also, morphological changes can also be indicators of stress, and fungal reproductive success. It is also a highly sensitive indicator. So this stress at individual level, for instance, is likely to reflect on the next level, that is the community level responses. Species richness, for instance, will decline and communities will shift. Community changes normally occur before we detect functionality loss, the litter decomposition, which also make them strong early indicators of ecosystem disruption before it reflects on the ecosystem functionality, which is our next level. If the stress persists at a community scale, for sure, we are going to start to see leaf litter decomposition capacity decreasing, consequently affecting the nutrient cycling and food web interactions. Here is just an example of a study where we observed that fungal-driven leaf litter decomposition slows down by 17% when in metal-polluted waters, and decrease even more, up to 24%, when combined with the second pollutant, in this case, the nanoplastics. This had a direct implication on the next trophic level, that is the macroinvertebrates. Here, the leaf consumption by the invertebrate reduced significantly when he was offered the leaves contaminated by the metals and the nanoplastics, especially at the highest concentrations. But while aquatic fungi are promising bioindicators, challenges remain. Fungal communities are diverse and interactions between species may be difficult to interpret. Fungal natural variability can mask pollution signals, and therefore standardized protocols are still needed. In this sense, future work should focus on expanding stressor coverage, for instance, emerging contaminants, develop functional indices and baselines based on the fungal responses, and as I mentioned before, integrate classical and modern technologies. In conclusion, I believe the take-home message here should be that aquatic fungi are crucial organisms in freshwater ecosystems, driving important processes such as the leaf litter decomposition and the nutrient cycling. Therefore, they can offer a sensitive and multi-level approach for detecting environmental stress, and their inclusion in biomonitoring frameworks, alongside other indicators, of course, could significantly improve freshwater ecosystem assessment and conservation. And that's it. I thank you again for the invitation, my supervisors in Portugal, the audience, and that's it. Thank you.
Thank you very much, Juliana, for this nice presentation. I guess it is clear that from your presentation, but also from the previous speakers, that aquatic fungi are so diverse and so important, but also that all these stressors that we find in the environment are very tricky and they are exposed to a lot of them. And this goes in line with a question that we have from the audience, which is, and I will make it right away, are some aquatic hyphomycetes adapting according to climate change?
Yes, in fact, temperature is a huge driver of aquatic hyphomycetes community in adaptation. Yes, so they are. I think, yeah, that's it.
Thank you. For the sake of time, there is other question, but I will ask that Juliana answers it by writing so that we don't take more delays. And I hand over to Bonnie to present last but not the least of our speakers today. Thank you.
Absolutely last, but by no means least. Next up, we're drifting to our last talk. Yes, I did that. It's Dr. Teppo Rämä. He did his PhD on Arctic marine driftwood fungi, hence the drifting joke. He's since been working with taxonomy, ecology, and discovery of bioactive compounds from marine fungi. He's an associate professor at UiT, the Arctic University of Norway in Tromsø, and is also associated with the Center for New Antibacterial Strategies at that university. Over to you, Teppo, to tell us about driftwood fungi.
Thank you, Moni. And while I'm sharing, I want to say Happy International Day of Biological Diversity. I think I got it quite close, IDB in short. I'm starting my presentation. You can see it now. Can you?
Yep, you're good to go.
Excellent. So my name is Teppo. I work in the north of Norway. And today I'm going to present to you give you a general talk about driftwood fungi, a bit how they look like, how they live, their ecology, and also the potential for using them in applied science and for biotechnological innovations. I included this picture here that's taken almost on the same date, 15 years ago, when I sampled my first driftwood pieces for my PhD. And I also included this picture here just to tell you that in this talk, when I talk about driftwood, I will talk about basically all wood in the marine environment, not only the logs that are drifting around in the sea. Okay, with that we start. So Patricia was a bit into the role of marine fungi, but I'm giving you a short summary here. So basically they can be found everywhere in the marine environment, and they play important ecological functions as symbionts of sustaining seaweed beds and seagrass, coral reefs, and also there are pathogens, for example, infecting diatoms and controlling phytoplankton blooms. We have saprotrophs that are able to degrade all kinds of substrates, including contaminated sediments, oil-contaminated sediments, and plastics. So remember, they are everywhere, and they are quite small, so they are not easy to find, but they are there. Okay, and because they have these ecological functions that are often mediated by chemical compounds, they have also huge potential to be, these compounds can be screened for biological activities, and of course there's a lot of undescribed chemical diversity that we can detect because these fungi are understudied. So the figure is a bit old already, but among marine organisms, fungi, and including the mangrove fungi, are the best producers, best sources for new chemical compounds. So up to date, it's already I think closer to 6,000 new marine natural products that are reported. So this figure is from 2020. And these new compounds include secondary metabolites that can be used in pharmaceutical applications, or at least has the potential for that. So like new antibacterial compounds, compounds with anti-cancer effect or anti-diabetes effect. And we also have enzymes that function well in low temperatures, for example, can be deactivated in quite not so high temperatures, not requiring so much energy. So they can be utilized in diagnostics. They are utilized in different types of industry, including clothes industry and for the production of biofuels as well. And as mentioned earlier, we have enzyme activity or enzymes that can be used in hydrocarbon degradation, so oil, and also bioremediation of heavy metals contaminated sites. And we have biosurfactants that can be used in cleaning surfaces. Okay, so important ecology, and that can, us humans can take use of that in different ways. I'm showing you a glimpse of how these fungi look like. So here you have nice pictures of mostly ascomycete fungi. So what is typical for these truly marine fungi, obligate marine fungi, is that the spores have often appendages. They have filaments, as Patricia showed, or gelatinous sheets, or different types of appendages that make the species and the spores float along in the water column, enabling long-range dispersal. This is a picture of a website that is quite useful when working with marine fungi. So currently there are a bit more than or almost 2,200 marine fungal species listed here. That's certainly not all, so this number is lagging behind all the time, and there is a lot of undescribed diversity. So the number of driftwood fungi is, I would say, somewhere between 1,000 and 2,000. I don't have an exact number for that. Here are some species occupying temperate driftwood. So on the left-hand side, you see the fruiting bodies that are like cushions or pads or flask-like fruiting bodies on the surface or often inside the wood. And in the right-hand side figures, you see some images of how the spores look like. We have, of course, lots of fungi in the tropics as well. Here is a book about mangrove fungi of Taiwan, including, I guess, several or some hundred species. Approximately, and we have also driftwood fungi or in deep sea. So here's a picture of a species that was fruiting in 5,200 meters below sea level. So really nice pictures of how this fungus is adapted to the high pressure in that environment. Really thick walls of the fruiting body, and you have special tissue there that is obviously strengthens the wall that it doesn't collapse in a high pressure. Yeah, so these fungi are mostly saprotrophs, meaning they decay the wood, and they can be found in natural wooden substrates, but in every man-made construction and processed wood that you put in the sea, sooner or later become colonized by marine fungi. And the fruiting bodies, as you see in the figures, they are often inside the wood. So you need to slice the wood in order to see them. And the structures that stick up are usually just a kind of a thin neck that protrudes from the tree. And through this thin neck, spores that are produced within the fruiting bodies are released to the seawater. Okay, and I've been working in the Arctic. You can see in the map, basically roughly my background, and the blue dot on that map is where this small movie is from. So this is from the island of Jan Mayen. You don't have the sound perhaps there, but it's waves and some birds, and you can see it's tons of driftwood. Although there is no wood and big logs, big trees growing in the Arctic, we have lots of driftwood there to study. And the condition is quite harsh. So when we work, it's usually, often it can be cold, especially in Svalbard archipelago. We have ice and snow and cold temperatures, so it's quite harsh to sample. And we also need to have rifles with us to keep scare off the polar bears if they show up. But despite all of these harsh conditions, people have fun, as here, like they are tilting on this swing on the big log while in the field. In my PhD, which I finished a bit more than 10 years ago, I studied 50 logs of driftwood using different techniques. And in those 50 pieces or logs, I found more than 900 species of fungi growing in there. So I didn't have so much time to focus on the morphological investigations and looking for the fruiting bodies, but I was able to find 25 species. I did a big culturing effort, culturing fungi out of wooden cubes, and got 143 species. And then using a next generation sequencing technique called 454 pyrosequencing, I was able to detect 807 operational taxonomic units that correspond roughly to species. And not so many of these the species found with different techniques were not shared so much. So there's a quite high diversity of fungi also in the Arctic driftwood, and certainly many species that remain undescribed. This phylogenetic tree shows my cultures, 143 cultures that I established, or isolates that I found in and established in my PhD. And now later years, I've been focusing on some of the groups here and working with the taxonomy and describing new species, and basically every kind of group I've digged a bit deeper into, also collecting more material and sequencing. I've been able to find undescribed species, and so far from driftwood, I think I have described 11 or so, and there are certainly more to come from this material. So quite high diversity again. Okay, but the biodiversity has its intrinsic value that we all appreciate. But of course, they provide us with ecosystem services and then also with biotechnological innovations. And I'm showing you a couple of articles that we have published in recent years. We have been focusing on antibacterial activity of marine fungi. And especially the paper on the left-hand side showed a really promising compound that was also killing some antibiotic-resistant strains of Staphylococcus aureus. So this is a promising field, certainly, and we will keep on looking into the antibacterial activity. Okay, I would like to end the talk here. Thank you, the project organizing this seminar, students, collaborators, of course, biodiversity, and you, and not least the people organizing the Driftwoodstock concert or festival up in Svalbard, because they gave me the inspiration for the name of this talk. Thank you so much.
Thank you so much, Teppo. Did you just say there's a Driftwood festival? Can we all go? That sounds amazing. I've got a quick question for you, and it's sort of in line with a question we had earlier, but with your Arctic driftwood fungi, are they threatened at all by warming climate in the Arctic? What's happening there?
Yeah, that's a good question. Certainly, I didn't explain it here because of the sake of time, but I have an extra slide on that. We'll see if we can jump to that. Continue to share. So you see that I said that the driftwood is not originating from the Arctic, so most of it coming to the western northwestern Europe and the Arctic shores. It comes from Siberia. And the big rivers transport it into the sea, and there it's caught by the sea ice. So, and then it travels many years to North Pole and caught in the ice. And when the ice melts, then these logs are released and they drift on shore. But now when the temperatures are rising also in the Arctic and really rapidly there, and we are losing the sea ice, so it means basically that I think in already, here it's in by 2060, I've seen also predictions for 2030, we will lose kind of new supply of driftwood. And then of course these species start to struggle, the species that are associated with the driftwood. Yeah.
Wonderful, thank you. And I think this next question should be really quick, and I'm also really interested in it, but that's from Anjali. Are driftwood fungi similar to wood rotting fungi?
To some wood rotting fungi. So these fungi are, we have in the marine environment, are pyromycetes. So, and they have same rot strategy than some of the terrestrial fungi. So there is certainly similarities. We see also some terrestrial species in these logs, and we know based on evolutionary studies that fungi have made the jump from between terrestrial and marine environments several times. And these fungi we find in the sea, in driftwood, they originate and diversified in terrestrial environment before jumping into the ocean.
Awesome, that's wonderful. Thank you so much, Teppo. And thank you so much to all of our speakers. I think there may be like the odd question that we couldn't address via typing frantically. We'll look through all of the recordings of the chats and the Q&As and try and provide as much detail in our follow-up as possible. Thank you so much. We had a really super attendance. I think at maximum was over 80 people on Aquatic Fungi, so that's very exciting. Obviously, it's International Day for Biodiversity, so please keep telling everybody that this day is happening today so that people understand that species are important, biodiversity is important, all levels of biodiversity, whether it's ecosystem, species, genetics, and so on. And also, obviously, then share this webinar link with all your friends, family, colleagues, people you pass in the street, people you meet in the pub, and so on, because it would be really nice to get more folks interested in this wonderful diversity that's literally all around us. Fungi are all around us, and as we've just learned, also very prolifically in aquatic systems. So that's wonderful. Thank you to all of my co-hosts and the organizations that helped with this. You've already got a lot of information of how to get in touch with us. We'll put that in the follow-up email again as well. And with this, happy International Day for Biodiversity. Keep celebrating and keep beating the drum for aquatic fungi. Thank you so much, everybody.